understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within the cell is more complex. Eukaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the histones are rich in basic amino acid residues like lysines and arginines, which carry positive charges in their side chains.
We have, yes, last class we learned about how packaging is done in prokaryotes. Prokaryotes, as we know, they don't have a definite nuclear membrane. So in the nucleoid, we call it nucleoid, right? Nucleoid. So in the nucleoid, how they are arranged? DNAs are negatively charged, and the proteins which are positively charged, in the diagram you have seen that in yellow colored proteins. Okay, the proteins were like this arranged, and on that proteins the DNAs were looped on. Okay, DNAs were looped like this, like making like a flower like structures. So that was in prokaryotes. Since prokaryotes don't have a definite nucleus, and even the chromosome number, DNA is all quantity wise, if you see it is less, so it is arranged in that way. I'm talking about prokaryotes. While in eukaryotes, so in prokaryotes, any doubts, children? In prokaryotes, they are arranged inside in the place called nucleoid. They are arranged, they don't have a definite nuclear membrane. So the positively charged proteins, around that positively charged proteins, the DNAs loop around and remains intact in nucleoid. That is not the arrangement in eukaryotes. In eukaryotes, DNA is arranged, what you can see just now, like this they are arranged. Okay, this arrangement helps, like this is the proteins, these are the protein, okay, that is a histone molecules, while these are, what you can see, this fiber like structure is nothing but DNA. Okay, further you'll be learning about it, how they loop around. Okay, so what you're going to learn is about, this is the structure, how they are arranged inside the nucleus. Okay, yes, we can go.
Prokaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the
This proteins up now, this we are talking about eukaryotes now, right? As you can see here very clearly, they have mentioned about the name of the protein also, it is here. Okay, here, that is lysine, adenine. These are the proteins, and proteins are positively charged depending upon the type of this kind of amino acid present in them. Okay, so we are talking about now histone molecules. Histones are protein, okay, made up of amino acid, and these are the names of those proteins, or amino acid rather. Okay,
histones are rich in basic amino acid residues like lysines and arginines, which carry positive charges in their side chains. Furthermore, the histones are arranged to form a unit of 8 molecules known as the histone octamer. Now around the positively charged octamer, a negatively charged DNA molecule is wrapped to form a structure known as the nucleosome.
Okay, we have understood that last class also, that DNA is actually 2.2 meters. With calculation, we have learned that DNA is in one cell. If we put it in a long lengthwise, we keep it. It around measures around 2.2 meters. Okay, and how then it is packaged? That is what we are learning. Fine. So this histone molecules, this spherical structures, what you see around octamers, means they are 8 in numbers. Okay, 8 octamers, this one we're talking about, they are the proteins. Okay, and this octamer means 8 histone protein. And how this, what you can see, the fiber like structure, this one is nothing but DNA. Now this also further we are going to learn how they come in this fiber form. Okay, that is later. Anyways, this is how this is DNA. It's rolled, wrapped around octamers, which makes it easy to be, and that is why it gives a look of, chromatin network. When you see any nucleus inside, other than the division time, that is mitotic or meiotic division time, the inside the nucleus, the condition of the chromosome or the condition of DNA, what you see is like a chromatin network, like this. Okay, and with some kind of like a beaded, like a beaded chain inside the nucleus, we find the structure of DNA. So that is what we are seeing now, how they are seen. Other than division time, majority of this time, this inside the cell, the condition of the chromosome in DNA is in this shape. Okay,
Which is held in place by the H1 histone.
now this is the what you, as at home, for clipping the cloth so that it should not move out in the wind and all. Parents, your mother use a clip, right? Same way, this is a kind of clipping. Okay, it clips. This is what, this is H1. Okay, and then this will what you call it, clip those DNA with histones and will not allow to scatter or move around. So it helps in a kind, I'm just giving an example that it works like a clip. Okay,
A typical nucleosome has around 200 base pairs of DNA helix, and the nucleosomes present in chromatin can be seen as a beads-on-string structure when observed under an electron microscope. It is
one
the
more thing I want to add is add in this here is, you can see each one of it is known as nucleosome, and inside that, this is the DNA, how it is seen. Okay, okay.
nucleosomes that make up the repeating unit in chromatin, the thread-like bodies present in nucleus. Now, the chromatin fibers are of two types: euchromatin and heterochromatin. In a typical nucleus,
Here you have to learn, okay, about two types of chromatin. Now, till now you have understood that it is packed inside, how it is packed inside. Okay, it is in the chromatin form, in this nucleosome, each what we call it as the octamer, that is a protein, along with the DNA negatively charged, get wrapped around, and with H1 proteins it is clipped, and like this kind of long chain, it is wrapped around and nicely remains inside the what you call it nucleus. Now coming to two types of chromatin. Okay, this is important to remember, because further you will come to know there are two types of chromatin, which out of which only in one we will be learning. One is very important, that is like euchromatins and all, where translation happens because of this euchromatin, the coding, which is very important, which type, that you are going to further learn. So please listen to it carefully about types of chromatin.
euchromatin fibers
Yes.
of 30 to 80 nanometers in diameter are loosely packed and stain light. On the other hand, heterochromatin fibers of about 300 nanometers in diameter
Okay.
are more densely packed and stain dark. Moreover, of the two types of chromatin fibers, euchromatin is transcriptionally active, while heterochromatin is inactive. These
Okay. So as I said, how you can make the difference between both the euchromatins, which are active in the sense the codes present in them helps in the transcription. The what is transcription? In one line, if I just conclude what is transcription, it is a process in which mRNA, messenger RNA, is made from DNA, the part of DNA which has the coding for various proteins, and that codes it passes it on to mRNA, so that it is a messenger RNA. It will move out of the nucleus and then it will translate into a protein. So this important process is done with the help of euchromatin, the type of chromatin which is present inside the nucleus, which stains light, and it is not that dense, it is loosely packed. So the how it is identified, when we stain them, it takes lighter stain, as well it is not dense, it is loosely packed. So that is how. And just opposite to antagonistically, we can say the character of heterochromatin, which is dark, it takes dark stain, and it is has thick kind of look when you see at them, it gives. And it is not this heterochromatin is majority number, huh? Euchromatin is very less in number, further you'll learn, while heterochromatin is quantity wise also they are more. As well, they are not active in the sense of transcription. Transcription is very important in our body, which helps in providing different types of synthesis of proteins. Okay, so transcriptionally they are not active. We are talking about hetero, while euchromatin are active and they take part, and they are the one which are coded for various proteins. Yes.
chromatin fibers coil further and condense to form short and thick bodies called chromosomes during the metaphase stage of cell division. In this way, a DNA double helix is packaged into a chromosome, which is further packaged within the nucleus. If we store Nevertheless, many biologists were still unconvinced that DNA is the hereditary material. And so the search for genetic material continued for several years and was concluded with the discovery of DNA. Once it was proved that DNA was the primary genetic material in the living system, it was natural to conclude that DNA replicates as a molecule, qualifies as genetic material only if it replicates. Scientists Watson and Crick, who in 1953 had proved that DNA had a double helical structure, had also suggested a probable scheme for the replication of DNA. According to their proposed scheme, the two strands
Okay.
separated
Before we move ahead, this is going to be the next topic now, replication. Okay, I just wanted to ask, is it clear? This small topics are getting over now. This is packaging of DNA is over. If you want me to conclude it in few statements, let me complete it. The packaging of DNA, since DNA is 2.2 meters, as we calculated with the number of bases and the distance between the two bases, we have calculated that it comes when we multiply it, we get 2.2 meters in one cell we are talking about. So how it is packaged in a very simple sentence, in 2-3 lines I am concluding it. Okay, it is uh, how it is, just once it is once it is this how that we are learning about, in few sentences I'm going to conclude so that it becomes easy for you to review. Okay, how the DNA is packed. DNA is a negatively charged component. Okay, so what it does, it has got 8 molecules of proteins, that is octamer of proteins, which is positively charged. Around that positively charged proteins, negatively charged DNA wrap around, while along with H1 histone, again that is a protein which will clip it to so that it remains, and that becomes one nucleosome. So they are nucleosomes, and this is the structure how they are. They remain in the nucleus other than the division time. Okay, other than the division time, this DNA remains in the form of chromatin network. Okay, and this chromatin network, and this chromatin network, and this chroma means you are also able to hear those sounds.
No, ma'am.
So these chromatin networks are two types in the nucleus. One is eukaryotic and euchromatin and heterochromatin. Euchromatin is the one which is active. Uh, they are active transcriptionally, while the heterochromatins are non-active transcriptionally. This is all what we learn in uh, the topic under packaging of DNA. Anything you want to ask? Else we will move to the next, that is replication. Nothing? Anybody?
No, ma'am.
Okay, so we'll move next to replication. Actually, replication means this is the first and the foremost quality of being, or to be called as, uh, a genetical material. To call it as a genetic or genetical material, it needs to have some features. One of the very important feature is be able to replicate, and that means to make the copy of itself. Okay, so and we are going to learn how DNA is able to do that. Okay, now during meiosis and mitosis, we have learned that half of its structure, half of the structure of DNA is during, like this. If you see, okay, half of it's like we have seen, like I'm not able to write. We have learned, like, if this is DNA structure, half of it, no, like this, half of it moves to one side, right? It moves to one side, the other half move to the other side during the meiosis and as well as mitosis. It happens, and then later on we learn that these moved half of this chromosome or DNA will make its other half of it. So we are going to learn how it makes the other half. Okay, that is what we are going to learn, and this is known as a replication or replicating itself. So the scheme was given by Crick, okay, was given by him, but then the thing is, what he said was later on proved with many experiments. What we are going to learn further, okay, is a replication of DNA, the first quality or the character or the feature a genetic material should have. Okay.
and acted as a template for the synthesis of two new complementary strands. After the replication process was completed, each DNA molecule would end up with one parental strand and one newly synthesized strand. That is why this DNA replication scheme was referred to as semi-conservative.
Okay, semi-conservative. About this, actually, this scheme, this was just a scheme first. Scheme means thought, okay, and then later on it was proved with experiments, which we'll be learning. Now, in that, what they said was, in this scheme, the scheme says that the DNA has a structure of a, like this, it has a structure. Okay, so what they say is during the replication, what happens is this kind of twisted turned DNA structure, the double helical structure, unwind themselves. Okay, the first step is they unwind themselves and form like a, you know, like this. They will unwind themselves, and that too in a small area only. They will, like, at a time this small area only has been unwinded. The reasons are very simple. This kind of replication process needs a lot of energy. So at a time, in on a DNA, only very little part, a fragment of it will unwind. Okay, and then this one will be the old strand, the other will be sorry, templates are there, two stands are there. Okay, this is also old, this is also old now. And we know that when they are moving out, if this is, I'm just giving one simple example, then we will start. If suppose, if I take the example of this, if suppose this is A, of course this was, here was T, if here it was G, say here it would have been C. Okay, this we know it. I'm just giving two nitrogenous bases only, I'm taking and explaining you. Now they have unwinded. Now when they unwind this old, now I will take only one strand of old template. Okay, so that, because I'm not able to make it so that, so if we see one strand here, as I said, one was A and the other, if I say it was suppose C, the other, this is old one. Okay, the old strand from where it has come. Now this old strand, by the nature of complementary pairing, we know that it will form a double hydrogen bond and will form here which nitrogenous base will come? T will come. Okay, that is thymine. While here, what will come? With a triple hydrogen bond, it will make a bonding with cytosine. And like this will be old, and one new strand will be formed here. One new strand will be formed, and that will be new strand. Okay, so since the complementary pairing they obey, so one strand is old and the other, the new one, will be made on the basis of the complementary pairing. Okay, and that's why this is known as semi-conservative. Semi means half, some of it. Okay, semi-conservative means one strand is always old and the other strand is always new. This is known as semi-conservative nature of DNA replication. Yes, we can proceed.
In 1958, to find conclusive evidence for this scheme of semi-conservative DNA replication, experiments were conducted by scientists Matthew Meselson and Franklin Stahl on bacteria Escherichia coli or E. coli, a bacteria that divides every 20 minutes. Meselson and Stahl grew E. coli in a medium that had NH4Cl15 isotope as the only nitrogen source. N15 isotope.
Okay. So if you remember the experiment in which DNA was proved, okay, that was Martian Chase experiment. Okay, in that experiment also we have learned that they have used the radioactive phosphorus and radioactive sulfur to prove that DNA, and that was accepted by all the scientists that yes, DNA is a genetic material. After that, now here you are coming with again little somewhat like that kind of experiment, in which what is the agenda, what is the motive? We want to find out that the scheme given by Watson Crick, that it has got the replication process happens in a semi-conservative way. That means one strand is always new, the other strand is always old. Okay, okay. That to explain, he has taken this experiment in which he took E. coli is a type of bacteria, and it is used for all these experiments, and it has the ability to reproduce within 20 minutes. Okay, so we can see the results very soon, after every 20 minutes. Okay, now what he did was he has taken and this E. coli and was growing it in the medium, that is nutritive medium, in which This is N15. Okay, the skin is very much zoomed, so we are not able to see N total. N15 type isotope of nitrogen, and this sodium. Okay, and N15. Okay, NH4Cl. E. coli was born. Yes, you can proceed. Time is only 10 minutes.
Is a heavy isotope of nitrogen, but it is not radioactive. Many generations of E. coli were bred in the same medium, which was not too hard to achieve since a new E. coli generation arrived every 20 minutes. As a result, newly synthesized DNA too had the N15 isotope as a constituent of purines and pyrimidines. This new heavy DNA molecule was distinguished from normal DNA by virtue of its high density while being extracted via centrifugation in a cesium chloride or CsCl density gradient. Later, E. coli cells were transferred into a medium with normal NH4Cl14 isotope. After the cells multiplied, their DNA was extracted and separated independently in CsCl gradients to record the density of DNA. It turned out that DNA extracted from the first progeny of E. coli after the transfer from the N15 isotope to the N14 isotope medium had an intermediate density. It was referred to as hybrid DNA. The next generation of E. coli, which was born after 40 minutes, had equal amounts of hybrid DNA and light DNA. Thus, the findings of the Michelson and Stahl experiment established the validity of Watson and Crick's.
Okay, now here, this is the experiment which proves about the semi-conservative nature of DNA. Now, what they did was, Michelson and Stahl, what they did, they took E. coli as they were growing it in the of nitrogen. Okay, now I told you, whenever you give the medium, from that medium only they will take the nutrition so that they can further multiply. Okay, so when they were doing this process of multiplication and growth by given in a medium, so what Salk and Michelson did was they provided with heavy isotope of nitrogen, that is N15 first they gave. Now, in two ways they are explaining this again. Okay, what they did was, first, if I say they have taken N15 and second time they took N14. Okay, now when cesium chloride is a medium in which they centrifugated the DNA and separated, and they saw the density of it, you can find it out. Okay, now N15, when we say it is heavy, N14 is comparatively lighter. Okay, now what they did, when the first time when they were grown, now when you see this red color, this is they are showing with the heavy, there is N15. So first, when if I say, if I also use the color blue and red. Okay, first when they have grown the DNA in the first generation, now what will happen, say suppose the DNA in the normal DNA is like this. Okay, I gave them N15. When I gave them N15, according to the scheme, what should have happened? One, they will segregate. Okay, for replication, this one of the strands will separate the old one and will come with one the other strand, and the other strand is, we are saying it is new. So if it is new, then it would be red. Okay, now these one blue and one red means it should be a medium. It should not have, when they have put it in the cesium chloride to check the what do you call it, the density of this, what they found, if suppose they were of this one. In the cesium chloride, in the books also you can see the picture. It is the heavy isotope will remain here. The position will be here according to its density. Fine. The what you call it, the medium, the heavy, the light one will remain here according to its density. While when they have made, they gave 20 minutes, and the second generation 20 minutes, 40 minutes, when they found the medium one, they found these DNAs were, the DNA were in the, somewhere in the, why it is going, huh? Somewhere in the, in the middle between the heavy and the light, they were somewhere here. So this medium density shows that one strand was heavy while the other medium. That you understand the density differences, children? Okay, in cesium chloride, when they did it and they found this kind of I mean, one old with one new strand was proved with this experiment of Michelson and Stahl. And we want to ask, as the time is also getting over, this is semi-conservative nature of DNA was proved. That
Theory of the
replication happens,
semi-conservative
but it is in a semi-conservative. That means the DNA strands, they are double helical. During replication, they unwind. See, the process we have not learned. This is only the scheme. We have to learn the process of replication, that is different. That in that you have to learn which enzyme is needed to unwind this much part of the, like suppose I told you, every time only a little part of DNA it segregates. Okay, so what is the enzyme which will come, and what is the next enzymes which will help in forming the